Hello everyone, welcome to IGLS Academy. This is your mentor Dr. Shamaita Basu, and in this session we will be covering the last part, that is the second part of the topic human gene therapy. Okay, so I think in the last session I ended with the different types of vectors, okay, which included the adeno-associated vectors to a larger extent, which are the most commonly used vectors as means of gene therapy, as means of vehicle for gene therapy. Okay, so in this slide, as you can see, basically it's focusing on how these AAV vectors, these adeno-associated vectors, are utilized for gene therapy. Okay, so let's focus on this diagram. As you can see, so this is basically the adeno-associated virus or the AAV vector that we have, which is containing our therapeutic DNA, means our therapeutic gene, which needs to be delivered to the targeted cell. Okay, now this is the scenario outside the body. While inside the body we can see that this virus is introduced or injected inside the body of the patient. So this is the cell of the patient. So after getting injected, or after like getting inserted inside the cell, it is obviously how it is inserted inside the cell, with the help of the endosome, which is a very important cell organelle. Okay, so gradually this endosome, after insertion, after successful insertion or incorporation inside the cell, this endosome will slowly dissociate or break down. Okay, and after that, gradually from inside that virus, the nuclear contents will be starting to come outside from the the cell of the virus or the virus particle. Okay, so this is the therapeutic DNA that is coming outside from the viral cell, and then this DNA forms a circular episome. Okay, and then it is getting outside from the body after protein synthesis, because the ultimate aim is that the therapeutic DNA will go inside the body of the patient. It will become a part of the DNA or the nucleus of the patient, and it will be starting to produce the protein that it is supposed to encode for. Okay, so this diagram is only explained over here step wise. So let's see, in the first step, as you can see in the diagram, the virus is taken into the cell via the endosome. So this is the first step. The second step will be the endosome is breaking down, as I explained. You can see also over here that the endosome is getting broken down. Third step, the therapeutic DNA enters the cell nucleus as a double-stranded molecule ready for transcription. So it is ready, already it is ready for transcribing, and then for translation to get the ultimate protein product. Okay. Then step number 4, as you can see over here, the therapeutic DNA will be forming a circular episome. And then step number 5, as you can see over here, upon promoter activation, the transcription will occur. Okay, promoter, again something I have talked about in details in many of the previous sessions. What is a promoter, what is an operator, what is a repressor? Okay, mainly when I talked about modulation of gene expression, when I explained about the different operons present in the prokaryotes. Okay, and last but not the least, number 6, the resulting transcript leaves the nucleus and travels to the ribosome for translation. Because again, this is something these are very important concepts that you need to have a clear idea about. Okay, that protein synthesis needs ribosome, and hence it needs to come out from the nucleus. Okay, because ribosome is present in the cytoplasm, it is not present in the nucleus. So just after replication or transcription, the polypeptide chain or the protein molecule that we have, it needs to come outside from the nucleus to the cytoplasm for translation, and most importantly for the post translational modifications. Okay, okay, now moving forward in the next slide, as you can see, it is a basically overview of how these AAV vectors are produced. Overview of the development and production of these AAV vectors, that is adeno-associated vectors. So it is basically a complex, complicated and a sophisticated process. Okay, so see, in the first step there will be transgene development. Okay, so what we have the cDNA, that is the complementary DNA, which is reverse transcribed from the human mRNA, and there is no introns. Okay, what are introns? Introns are the intervening sequences, means the non-coding sequences. Okay, so the cDNA that we will have, it will not be containing any introns, it will only be containing the exons. Okay, so this is the vector genome that we have. Okay, so this is the transgene over here. So from there we need the AAV helper, because adeno associated virus. Why? Because it is needing the help of another virus to act as a vector. So that's why it is also known as the helper. Okay. So this is then transfected inside the cell. So this is the E1 expressing cell, and then it will be producing the cell line, and finally gradually after several several rounds of producing the cell line, we will get the AAV variant. So basically there are three steps: transgene development, then comes the AAV GRT production, and then comes the concentration and purification, and last but not the least, quality assurance, which is very very important. Okay, so if you are producing the AAV-GRT, that is not sufficient. Okay, so the AAV-GRT means the adeno-associated virus gene replacement therapy. Okay, so if only you are successful till the second step, that is not enough. The third step is very much important and essential, because we need to know about the concentration and the purification of the AAV vector that we are producing. Okay, if the concentration and purification is not satisfactory, then just only production will not help us. Okay, and also quality assurance, where the quality of the vector is perfectly okay or not, that needs also to be tested before we move on to the actual gene therapy. Okay, so purification produces vectors with high titer, high potency, and high purity. High titer means high load. Okay, high potency means high potency as like having a high potential to act as the vector in terms of the gene therapy or gene delivery. Okay, and also high purity, it needs to be very much pure. Okay, if there is any kind of contamination with any kind of other virus or fungus or bacteria, then that is again something very much unwanted, because it will be going and producing more complicated conditions and diseases and infections inside the body of the patient. Okay, so that is again something unwanted. And also QA using FDA established requirements and predetermined specifications. Again, quality assurance. Quality control and quality assurance is very, very important, not only in case of vector production, but also in case of production of any new drug or medicine or vaccine. It is a very important part of the entire process, quality control and quality assurance, which is done in accordance to the specifications and regulations given by FDA, that is Food and Drug Development Association. Okay. Okay, now moving forward, so gene therapy is a novel technique which uses manufactured transgenes to address the underlying genetic cause of a disease. Gene replacement therapy or GRT aims to provide sufficient gene expression in enough targeted cells to ameliorate or correct a dysfunctional phenotype. Okay, again, what do you mean by a dysfunctional phenotype? Phenotype means something which is physically visible, which is appearing from the outside. You can see it from the outlook or the physical appearance of that particular organism. Okay, so in order to correct or rectify any kind of dysfunctional phenotype, we need to accurately target the cells which needs to be corrected in order to ameliorate that particular condition. Ameliorate means to cure or to treat. Okay, recent accomplishments in gene therapy techniques have advanced safety and efficacy with viral vectors. AAVs are one of the most promising viral vectors for gene transfer and expression. I think this is something which I have already mentioned in the earlier session. Okay, now I have given a few examples of several somatic gene therapies which are still under investigation. Means they are still under the research level. Okay, still under research. So firstly we have endothelium. So genetically altered endothelium can secrete a needed protein directly into the bloodstream. Okay, so if there is any problem with a protein which needs to be which needs to be, or which usually is secreted from the endothelium, then we can also go for a gene therapy which will directly produce the proper or the expected protein, and release it into the bloodstream. Next we have skin. So skin cells grow well. A person can donate a patch of skin after a genetic manipulation. The sample can grow to the size of a bat mat within 3 weeks, and the skin can be grafted back onto the person. Skin grafts can be genetically modified to secrete therapeutic proteins. Okay, so now when we are talking about skin, skin is something which is very fast growing. Okay, and skin grafting means, now grafting can be of various types. We can take skin from the same person and give to the same person, to the like to any other part of the same person which they are talking about over here. Okay, so if there is any genetic abnormality related to the skin or any other organ also, we can just take a patch of the skin, we can insert the therapeutic gene inside the skin, and then it can be replaced, or it can be placed into that region of the body of the patient where the problem is going on. Okay. Next, we have muscle. Muscle comprises about half of the body's mass. It is easily accessible and is near a blood supply. Treatments for muscular dystrophies can be very ideal, because muscular dystrophy, again, it is a kind of a genetic disorder, or you can say it is a type of a autoimmune disorder also. Okay, muscular dystrophy, where the normal muscle contraction and relaxation ability of the muscle fibers are lost. Okay, it's basically known as Duchenne muscular dystrophy. The entire name of the disease is Duchenne's muscular dystrophy. Okay, so it is a condition where we can actually utilize the muscles, because muscles are again something which are present all over the body, isn't it? The entire body mass, we have muscles. We have muscles, we have proteins, we have fat, and we have skin. That all together makes up the entire body mass, the lean body mass that we know, except the water weight of the body. So, and also not only that, it is also another important factor is that muscles are having a great amount of blood supply. Okay, so again it is something which is preferable in terms of gene therapy, preferable candidate for in terms of gene therapy. Okay, then we have liver. Again, we all know it is the largest organ of the body, though some say that skin is the largest organ, but ideally skin cannot be referred to as an organ. Like, it's a disputed kind of a topic. Okay, some say that skin is the largest organ, and some say that liver is the largest organ. Okay, because skin is something which is present entirely throughout the body. Now a specific organ ideally cannot be present throughout the body, isn't it? Organs means there are specific masses of tissues which are present at specific parts of the body, and they are performing specific functions. So in that perspective, if we think, then obviously liver is the largest organ of the body, and also it is the organ which is having the highest regenerative capacity. Okay, means if you just cut off half the liver of a person, of a healthy person also, disease person, so obviously of a healthy person also, if you just cut off half the liver, after few months the entire liver will be grown back. So that means that half liver which is left behind, it will regenerate itself and form the remaining half of the liver, and within few months we will get the entire complete liver again. So that means the regenerative capacity of the liver is very high, and that is why it can be a very much suitable candidate for successful gene therapy trials. Okay, so it is the largest organ. It is an important candidate for gene therapy because it has many functions and can regenerate, exactly what I said. To treat some inborn errors, at as little as 5% of the liver's 10 trillion cells would need to be corrected. Okay, so the liver consists of total, say for example, 10 trillion cells. So among them, if we can regenerate at least 5% of the cells, it will be sufficient for us to treat such diseases. Okay, what kind of diseases? Mainly the inborn errors of metabolism. Again, something I've talked about in the previous many sessions and topics, like we have phenylketonuria, we have alkaptonuria, then we have glucose 6-phosphate deficiency. Okay, so these are some of the inborn errors of metabolism, where some of the main enzymes which are involved in some of the major metabolic pathways are actually absent in the body or produced in the body in very less amounts, and that leads to various problems or various defects in the different metabolic and biochemical pathways of the body. Moving forward, we have lungs. So lungs means obviously the respiratory tract. It is easily accessed with an aerosol spray. Several aerosols to treat cystic fibrosis replaces the defective gene, but the correction is short-lived and localized. Okay, so cystic fibrosis, again, it is a genetic disorder. Okay, where there is continuous respiratory distress, breathing problems, and it in many of the times it leads to COPD, that is chronic obstructive pulmonary disorder, which is in many cases non-reversible, and it can also lead to death of the patient. Then we have nervous tissue. So nervous tissue, we all know that the structural and functional unit of the nervous system are the neurons. Okay, so the neurons are difficult targets because they do not divide. Neurons and RBCs and the brain cells, these are some of the cells of the body which do not multiply or which do not divide. Okay, gene therapy can alter other cell types such as fibroblasts to secrete neural growth factors or nerve growth factors, or manufacture the enzymes which are necessary to produce certain neurotransmitters like acetylcholine. and then the altered cells will be implanted. So basically we are doing all the manipulations outside the cell, and then we are just inserting the cell inside the body, because in general the neurons are the non-multiplying or non-dividing cells of the body. And last but not the least, we have cancer. So about half of current gene therapy trials target cancer, because cancer is again the same thing I need to tell over here. It's a kind of repetition you can say. So cancer is a disease which can occur in each and every part of the body. There is no part of the body which we can say that okay इधर तो cancer नहीं होगा. ओके ऐसा कोई भी पार्ट बॉडी का नहीं है. इन ईच एंड एवरी कॉर्नर ऑफ द बॉडी अ पर्सन कैन डेवलप कैंसर. And again also cancer is a type of a disease which mostly has a genetic predisposition. Okay, so that's why for when we are talking about gene therapy trials, cancer will be ideal, any type of cancer will be ideal disease where we can go for gene therapy trial. Okay, so these approaches enable the cancer cells or their neighbors to produce proteins that dampen the oncogene expression, bolster the tumor suppression, strengthen or redirect the immune response, or induce apoptosis. Okay, now what are these terms? What are oncogenes? First of all, so basically oncogenes are formed from pro-oncogenes. Okay, and what are not pro-oncogenes, proto-oncogenes. Okay, and what are proto-oncogenes? Proto-oncogenes are those genes which are involved in normal cellular growth and division, or you can say that they are involved. If proto-oncogenes are present in our body, then that means we are safe from cancer, because they are promoting normal cell growth and development instead of abnormal cell growth and development. Okay, now, when these proto-oncogenes are overexpressed, they are converted to oncogenes. And these oncogenes are now involved in abnormal cell growth and division, which is exactly what happens in any type of cancer. Uncontrolled or abnormal or, proliferative cell division. Okay, uncontrolled expansion or uncontrolled proliferation of cell growth, something which is not planned or maintained. Okay, so those are the oncogenes. So it is very important to dampen, means to slow or to inhibit the expression of those oncogenes in order to inhibit or prevent cancer. Okay, then we have tumor suppressor genes. So tumor suppressor genes means also, from the name only we can understand that those are the genes which are involved in suppressing the expression of the tumors, and tumors are something which can develop or which can lead to cancer. Okay, if it's a benign tumor, then okay, but also benign tumor can sometimes go and develop itself into a malignant tumor, and if it's a malignant tumor, then that means sure shot it will cause cancer. Okay, so tumor suppressor genes are those genes which needs to be expressed at an optimal level in order to prevent cancer, isn't it? And then strengthen or redirect the immune response, because again when any type of cancer is there inside the body of a person, his or her immune system like has gone for a toss, or his or her immune system is not working properly. Okay, any type of cancer. So that is also there, that is also will be one of the motives of treating cancer. And also inducing apoptosis. Now what is apoptosis? Apoptosis is normal cellular death, or you can also refer it as programmed cell death. Means cell death which is normal, which is physiological, which is not pathological. Okay, inside our body each and every cell has a particular lifespan, and after that particular lifespan that cell undergoes death. That is what is programmed cell death. Okay, or apoptosis, and it is a very important aspect for the normal physiological functioning of the body. And in case of cancer, what happens, this apoptosis is not happening normally, which leads to cancer. Okay, because after some time what happens, normal cells they wear and tear, isn't it? After a normal lifespan the cell has to die. Now if there is a cell which has to die but it has not yet died because of some defects in apoptosis, then there are chances that that dead cell or that defective cell, because the cell has done its function, isn't it? It has done and dusted, so now it has no longer function to do, but still it's remaining in the body, it has not yet been dead. So that means it is a prospective candidate, or it is a probable candidate to cause some type of infection or to cause some type of problem inside the body, which can be cancer. Okay, so inducing proper apoptosis at proper time will also be an important aspect of treating cancer. Okay. Okay, now I have given two important success stories of gene therapy. Okay, so one is treatment of Canavan disease, which is a neurological disorder. I will be explaining via the diagram, which I will be showing in the next upcoming slide. So it was seen in case of Jess Gelsinger's एक इंसान था उसके it was seen in case of that person. So Canavan disease is an ideal candidate for gene therapy for several reasons. What are the reasons? Number one, the gene and protein are well known. There is a window because whenever we are coming up with a gene therapy for a particular disease, it's very important that we know details about that disease. We know how that disease is causing, we know about the prognosis, we know about the treatment or the management methods, we know about the prospective candidates which can suffer from that disease. Okay, so it's very important to have a detailed idea about the entire etiology, the prognosis, etc. of the disease. Okay, so second is there is a window of time when affected children are healthy enough to be treated. Number three, only the brain is affected because, as I said, it is a neurological disorder. Brain scans can monitor response to environmental treatment, and next is no traditional treatment exists. So this is a diagram which is showing a baby, which is baby or a child who is suffering from Canavan disease. So this is a typical phenotypic outlook of that particular patient, as you can see in the diagram. So the name of this person, of this boy child, is Max Randall, and he was expected not to survive the first 2 years of his life. Today he is on the brink of adolescence thanks to gene therapy. So he has survived and he has also reached his adolescence, means around 13 to 14 years old. So this is exactly what happens in case of Canavan disease. So it leads to brain atrophy. Okay, means the brain cells die. And it also kills the oligodendrocytes. So here we have the oligodendrocytes and here we have the neurons. Both are important parts of the nervous system of the brain and the spinal cord. Okay, and neuron, we all know that it's a structural and functional unit of the nervous system. So this product is getting accumulated inside the brain, that is N-acetyl aspartate. Why? Due to deficiency of this enzyme aspartoacylase. Okay. Uh, why aspartoacylase deficiency is occurring? Because this aspartoacylase gene is not being expressed inside the cells of the neuron. Okay. As a result, it's leading to absence of this enzyme which is supposed to convert this N-acetyl aspartate to some other intermediate, to some other metabolic intermediate. So since this enzyme is absent, okay, see here stop is written, means this enzyme is no longer there, no longer getting produced inside the body. As a result, this N-acetyl aspartate is no longer getting converted to its intermediate metabolite, and it is getting slowly accumulated inside the brain, which is leading to brain atrophy and also killing the oligodendrocytes. Now, there are some important scientific issues which we need to know about when we are talking about gene therapy. What are they? Number one, which cells to be treated and how? Number two, what proportion of the targeted cell population must be corrected to alleviate or halt progression of the symptoms? Means we need to know about the correct age group. Is overexpression of the therapeutic gene dangerous? Because in many cases, overexpression can be beneficial. In many cases, overexpression can be harmful. Okay. Is it dangerous if the altered gene enters the cells other than the intended ones? Okay, means except the target cell, if it is going and entering into the other normal cells of the body also, will it cause trouble or will it be okay? So these are the things that we need to know. Then how long will the affected cells function? Will the immune system attack the introduced cells? Again, a very important point. If the immune system is attacking and going the introduced, going and attacking and going inside the introduced cells, then obviously that is something which will be causing problem inside the body of the patient, isn't it? So in that case, we need to immunocompromise that patient by giving immunomodulators. So these are important perspectives or aspects that we need to keep in mind. And last but not the least is the targeted DNA sequence in more than one gene. Means if the disease is caused by alteration in only one gene, or maybe multiple genes are involved in causing alteration of that particular product. And there are some bioethical considerations also. What are they? Number one, does the participant in a gene therapy trial truly understand the risks? These are basically the ethical concerns or the ethical issues which I will be talking about in the last topic of this course of medical biotechnology, that is societal issues. Okay, so there I will be talking in details about the different ethical issues. Second is, if a gene therapy is effective, how will the recipients be selected, assuming it is expensive at first? Because in a country like India, which is still poor and developing country, it's very important to first consider the financial background or the economic background of the patients that are coming, whether they will be able to afford it or not. That is also a very important aspect that we need to keep in mind. Third, should rare or more common disorders be the focus of gene therapy research and clinical trials? What effect should deaths among volunteers have on research efforts? Should the clinical trials be halted if the delivered gene enters the germline? Because germline में enter किया है मतलब it will be inherited to the parents, to the offsprings or to the next upcoming generation. Now these are some of the diseases where gene therapy has been found to be successful. Okay, and these were conducted between the year 2009 to 2011, so many years ago. So one is retinal disease, which is also known as Leber's congenital amaurosis. It causes blinding. The person will not be able to see anything. Then we have X-linked SCID, that is severe combined immunodeficiency disorder. Then we have ADA SCID, that is adenosine deaminase deficiency SCID. Then we have adrenoleukodystrophy, Parkinson's disease, and this has led to renewed interest in gene therapy after this successful attempts. Okay. So in case of cystic fibrosis, we use adenovirus selected vectors, okay, which are non-integrating, replicative defective, and respiratory virus. So there have been 3 research teams consisting of 10 patients or teams who had undergone these gene therapy trials for cystic fibrosis. So 2 teams administered virus via aerosol delivery into nasal passages and lungs, and 1 team administered virus via nasal passages only. Only transient expression was observed because the adenovirus does not integrate into the genome like the retroviruses. So that is the basic difference between adenovirus and retrovirus. Okay, then we have familial hypercholesterolemia. What do you mean by familial? Means it's genetic. It's coming from the other members of the previous members of the family, means from the parents or the grandparents. That is familial. And hypercholesterolemia means, see, anything which is ending with the term amia, with the letters amia, means it's referring to the concentration in the blood. Hyper means increased and cholesterol means cholesterol. Okay, so means increased level of cholesterol in the blood. So that is what is familial hypercholesterolemia. So it occurs due to defective cholesterol receptors on the liver cells. As a result, it fails to filter cholesterol from the blood properly. Cholesterol levels are elevated, increasing the risk of heart attacks and strokes. So the first attempt to treat familial hypercholesterolemia via gene therapy was done in the year 1993 using a retroviral vector ex vivo. Okay, and it was infused back into the patient and improvement was seen, and since then many different trials have been done. Okay, now we can also use stem cells for gene therapy. What are stem cells? Stem cells are a particular type of cells which can differentiate and develop into any type of other cell. Okay, so that is the unique feature of stem cell. So a stem cell can give rise to a liver cell, it can give rise to a stomach cell, it can give rise to a human cell. So basically they are having multipotency. Okay, potency to develop into different multiple types of cells. So now stem cells can be of different types. Over here we are talking about the HSCs or the hematopoietic stem cells. Means those stem cells which give rise to the blood lineage cells, to the different types of blood cells like RBCs, WBCs and platelets. So they will be ideal candidates for gene therapy. Why? Because they are self-renewable. Hence repeated administration of gene therapy can be reduced or eliminated, and they can be easily isolated from blood, bone marrow and umbilical cord. These stem cells, these hematopoietic stem cells, they can give rise to myoblasts, means the muscle cells when they are injected into the muscle tissue. They can give rise to the neural stem cells when they are, and therefore they can be, potent or potential candidate for treating gliomas. And we also have the embryonic stem cells which provide maintenance for therapeutic effects rather than the adult stem cells, because embryonic stem cells are much more potent, or they are having that multipotency at a higher level than the adult stem cells. Okay, because the embryonic stem cells means they are being isolated from the embryo, so they are having a much more potential to differentiate and develop into different other types of cells. So I think that's it about the human gene therapy. So we will soon be moving on to the next topic of this course, that is medical biotechnology. Till then, thank you, goodbye. Please like, share and subscribe ITLS Academy.